This episode is brought to you by Progressive Insurance. Do you ever think about switching insurance companies to see if you could save some cash? Progressive makes it easy to see if you could save when you bundle your home and auto policies. Try it at progressive.com. Progressive Casualty Insurance Company and Affiliates. Potential savings will vary. Not available in all states.
As a small business owner, sometimes it feels like no matter how much planning you do, there's always surprises, like an urgent, expensive repair. But here's a surprise you will like. With Progressive, small business owners save 13% on their commercial auto insurance when they pay in full. So enjoy a surprise for once. Get a quote in as little as 8 minutes at progressivecommercial.com. Progressive Casualty Insurance Company and Affiliates. Discounts not available in all states or situations.
Support for this podcast comes from Progressive, America's #1 motorcycle insurer. Did you know riders who switch and save with Progressive save nearly $200 per year? That's a whole new pair of riding gloves and more. Quote today. Progressive Casualty Insurance Company and Affiliates. National average 12-month savings of $197 by new customers surveyed who saved with Progressive between October 2024 and September 2025. Potential savings will vary.
Do you like sea stars? I call them starfish, but who doesn't love them, right? Did you know that on the Pacific coast of North America, we almost lost all of them? In today's episode, Dr. Ian Hewson joins me. He's a marine microbiologist and a professor at Cornell University, specializing in ocean virology and microbial ecology. I know you're sitting there laughing. Those are big words for me. Now, let's get back to the sea stars. In 2013, what became known as the sea star wasting disease, it began all along the entire Pacific coastline. It was one of the most devastating marine epidemics ever recorded. And we're talking deformities, loss of limb coordination, tissue decay, and just so much more. Billions of sea star populations were lost. And Ian, he was in the thick of it. He and his team studied it, trying to find out what the cause was. And we get into what they discovered, what was associated with it, and so much more. Lately, Ian's moved on to sea urchins, some other really cool sea life. This episode is about these beautiful sea creatures we love, the fragile balance of nature, the effects of the environment, and the list goes on and on. So sit back and get ready as we go all the way to the microbes in this one.
For real, 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 for real. Got a bunch of furry friends and I love them all the same. I'm an animal lover, veterinarian is the name. Let's share some pet stories from heartwarming to strange, losses and gains, from wacky to insane. For real. You're now tuned in to For Real with your host, Mark Kyle.
Hey everybody, since we did the recording, a major event happened and I've got Ian back on. I'm interrupting the episode because this is an important message. And just for you guys that listen, you know, we're all animal lovers out there, but it takes people like Ian and young students as well to do that. And there's been a big loss, and anything you can do to help, I would appreciate it on my end. And if you need information from me, you can always email me at mkyle@freepodcast.com and I can give you more information. But I'm going to pass the baton to Ian to let you know what's going on. And again, please help out if you can.
Thanks, Mark. Yeah, no, unfortunately, since the recording of the episode on Sunday, the 3rd of May, there was an absolutely catastrophic, devastating fire at the College of Marine Science at the University of South Florida. This is an institution which has been around for about 60 years. It's led the way for a long time in monitoring the Gulf of Mexico. It's had some significant scientific contributions, and about 60% of their labs were lost as part of this, completely burnt to the ground. As part of that, unfortunately, a lot of people have lost personal effects, laptop computers, iPads, iPhones, you name it, anything. If your office is like mine, it's full of stuff that we have as sort of mementos of our research. So in addition to losing all of the samples, the data, et cetera, from many, many decades of work, they've also unfortunately lost all of their personal materials for doing their work as well, things like computers. So just appealing to anybody out there, if you'd like to support their recovery efforts, there's two ways you can do it. The first is a general fund at the University of South Florida College of Marine Science. That goes directly to the institution. They will use that to support rebuilding the building, rebuilding their research capacity. And the second is specifically for the graduate students who are out there who have lost so much in this fire, and help them to rebuild and get back on track with their studies. So anything you can do to help, would really appreciate it.
And then just for you guys that know, the link and information will be in the description. But again, if anybody's ever lost anything, I've actually had a fire at my house one time, or if somebody broke into your car and stole something, you know how that feels. It's pretty painful. So you've got these young students are going through there that are on limited budgets, as I was just saying earlier, the macaroni and cheese budget. These guys have lost a lot too, aside from all the information and the studies that. So anybody that can help out, I would greatly appreciate. And now we're just going to get back to the episode, but I really do appreciate everybody listening, and hopefully some of you guys can help out, and I really appreciate it. Thanks so much. Hey, everybody, and welcome back to the For Real Podcast. Today, my guest is Dr. Ian Hewson. I'm sorry, guys. I'm having a slow time here today. We're going to have this is going to be a fun one for me. And there's a lot of reasons why. I grew up on the coast of California with tide pools and all that. But your expertise is something that I find really fascinating. I'm really excited for everybody to get to meet you. So welcome to the show.
Thank you for having me. Really appreciate it. It's great to be here. Great to talk to everybody. Thanks for the audience for tuning in.
Yeah, and I think this is a perfect audience for what you do. You're a scientist and do some really cool things, and we're going to get into some of that too. But like I always talk to most people when I start, and people that listen, I really like to get into who you are. So I kind of want to talk about your journey and move all the way forward. But let's go as far back as you want, as a kid if you want to, or whatever. I'm assuming you've always been kind of that science nerd, really cool guy. So wherever you want to go, let's go for it and hear your story. So you go for it.
Yeah, no, for sure. It's been a part of my life for a super, super long time. So I actually was born in Singapore
cool.
long time ago, back in the dark ages. And growing up, I grew up in Washington, D.C., where my dad was working for the United Nations. He was an agricultural scientist. And so he sort of instilled at a very early age kind of the scientific method, and we're doing science projects with controls and treatments and things. And this is when I was all of like 8 years old. So that sort of drew me into science at a very early age. And then, I moved through sort of high school. I actually moved to Australia, to Canberra, Australia. And I'm from Australia because I say Australia, not Australia.
Yes, I caught it.
I do slip in a few Australianisms here and there and get weird looks from people when I do it. But and I spent a long time in Canberra from high school, and then I went to university at the University of Queensland up in Brisbane. And pretty much I became interested in marine science when my dad used to take me sailing off of Hilton Head when we lived in Washington, D.C. And we used to see manta rays jumping outside the boats, and
that's cool.
Used to go out just catching shrimp with like cast nets and things. Used to go out flounder fishing, and really kind of was drawn to it, and used to watch Jacques Cousteau on TV. I mean, that was definitely kind of a formative TV show for it. And then so when, high school came around, they did all sorts of career counseling. I did the questionnaires to sort of ask, like, what would you be good at? And it said I'd be good at driving cranes. That was my one. And I was like, well, it doesn't help that I'm afraid of heights. So that definitely wouldn't have worked out too well. I convinced myself I was going to be an anesthesiologist because that was the highest income earner in Australia at the time. And that was sort of what I had my focus on, was like, how am I going to make a living? And then I sort of did my first year of undergraduate at Australian National University in Canberra, and had with the aim of doing veterinary science. So working with animals, small animal, farm animals, that kind of thing. And I didn't get in. Because my scores weren't as good as what they should have been. I wasn't a huge academic achiever as an undergraduate, or actually beyond, to be honest. But what I sort of decided to do was marine science. And immediately I just kind of launched into it. I moved to the University of Queensland after my first year. I took every opportunity I possibly could to get in the water. I was a big diver at the time. I've been diving since I was 11 years old.
Wow.
I became a dive master when I was an undergraduate. I love being underwater. I love just the fact that you're immersed in water, surrounded by life in all directions, and it was really it's where I feel the most comfortable. I have a big affinity for the water. I've been a swimmer since I was like 10 years old. I play water polo. I enjoy boating. I do all the things related to water. And so at undergrad, when I was an undergrad, I sort of started off convinced that I was going to work on, big things, like fish and sharks and whales and that kind of thing. And then working with a mentor there, I became really interested in pollution, particularly sort of sewage pollution in the coastal zone and how that influenced sort of marine life. And initially, it was just kind of working on, seagrasses and algae, big kelp-like algae. But then I sort of became interested in kind of smaller things and microorganisms and how they were responding to this sort of pollution and how they were remediating it, how they're making it better. And so that's what I sort of focused on for what's called an honors year, which is kind of like a one-year master's degree in Australia. And then I got the offer to move to the U.S. and do a PhD by somebody at the University of Southern California, my wonderful mentor, Jed Furman. And so I moved over there to Los Angeles. It was a big shock moving from Brisbane, Australia to Los Angeles. Can't say I'm really a big fan of LA. Sorry to LA listeners, but at the time...
I went to high school in LA. I know. I know.
It was hard surviving on $11,000 a year there, but
SC, everybody's got money at that school.
Yeah, and well, maybe not the grad students, but certainly everyone else does. And it was 4.5 years of really hard grad work. We were doing oceanographic cruises. I went out to sea, 13 times or something in that time in 4 years, and these were cruises of 10 years. That'd be fun. 10 days or more, and we were out between port calls and things for a long time, and really just enjoyed being out there on these UNOLS vessels, University National Oceanographic Laboratory System. These are naval vessels which are run by the Merchant Marine, and scientists go along and do oceanographic work. So I was then working on microbes, and then I got this offer to move to Cornell, which is where I'm at right now in the College of Agriculture and Life Sciences. And I started off sort of thinking along the same lines as what I was doing with my PhD, which was, just working exclusively on the microbes and not anything else, involved with them. And then what really kind of got me interested was the roles of these microbes and actually, looking at their diseases that they cause. Almost all microorganisms in the ocean are not causing diseases. 99.999% of them. They do important things. Those ones that don't cause disease, they're driving nutrient cycles. They're creating oxygen that we breathe. They, make the oceans function, but they're not causing diseases. But that really small proportion that do cause diseases, boy, do they have dramatic impacts upon organisms. And basically, I sort of started my career in marine diseases, looking at, characterizing the different types of microbes that were out there associated with diseases, particularly viruses. That was my big thing at the time because it was completely untapped. Nobody had looked before, and we had some new approaches that allowed me to do it. And then as part of that, I started to look at echinoderms, which are this group of spiny-skinned organisms, so sea urchins, sea cucumbers, sea stars, crinoids, feather stars, I think they're called. And what was really interesting about them was that nobody had even looked ever at any kind of virus associated with any echinoderm at all. And it was kind of a big open book. And so I, got my hands on some specimens of urchins from out in Hawaii and looked at the type of viruses, and we discovered some brand new types of viruses that were infecting them, or at least were occurring in their tissues. And, that is basically how I came to be working on echinoderm diseases. It was, shortly thereafter, we had this thing called sea star wasting disease, which started in 2013. And I got drawn into that because I was pretty much the only person that knew about any of the pathogenic microbes associated with it.
So I want to interrupt you real quick. I'm going to go back and forward. So, but as I forewarn everybody who listens, my brain works all over the place. I'll get back to where we're at. So when I first went, this is relative but not. So are you a Vegemite eater?
I love Vegemite.
I know, it's crazy.
That's an Australian, like if you're an Australian citizen, you have to love Vegemite. But there's a trick to it. You have a tiny little scraping of Vegemite with a lot of butter on bread. You don't put they do this with tourists all the time in Sydney and elsewhere in Australia. They give you a teaspoonful of Vegemite. And it's the most disgusting thing in the world when you have that.
It is.
But a little scraping.
Oh, let me give you my little story. So I played rugby for the U.S. team. I think we may have talked about this when we first talked. Oh. But anyway, so my very first time going over there was going to play rugby. And actually, I think the first time I went over there may have been in the first World Cup, which was in Australia and New Zealand. But on the airplane, I'd never even heard of this stuff. On an airplane, they gave you it's kind of like, for you guys that are listening, think of when you go to McDonald's, you get French fries and you got those packets of ketchup. Well, they got the packets of this green, greasy, looks like grease packets of these things. I'm going, what the heck is this stuff? Really green. It's a protein. I mean, whoever marketed it great. But it's the worst. It's the worst tasting. It's the most disgusting thing I've ever seen in my life. It always cracks me when I talk to people from down under. They love it. I don't get it.
Yeah. Oh, it's good stuff, and it's absolutely full of vitamin B nutrients too. So
yeah.
It works great against mosquitoes. If you didn't know that.
Oh, really?
Yeah.
Well, maybe I like it better.
Yeah. No. Have some Vegemite every day, and it'll keep the mozzies away.
That's funny. So there's my first one. Now the other thought I had, because like I said, I grew up in Southern California. I grew up in high school. I was in Palos Verdes, where all the tide pools and stuff were. Awesome. But and I'll get to the tide pools in a second. So when you guys were going out on your voyages or your whatever you call them, excursions, were you going over to like Catalina and San Nicolas and all those islands there? Is that what you're doing?
Somewhat. So, I mean, we used to have a time series project, which saw us go out every month and stop halfway between Catalina and San Pedro. Mm-hmm. And it's a time series which continues till this day, and basically learning about like how things change over time in the ocean, what their drivers are.
Right.
But a lot of the cruises that we went on, so we did cruises from Barbados to Cape Verde in West Africa.
Oh, wow.
We did a lot of cruises in the Amazon River plume, so out of Barbados going south.
Oh, that's awesome.
We also went from, Hawaii out to Midway and back, and even the South Pacific from Australia to New Caledonia to Fiji to Hawaii. Oh. So pretty much all over the place. But it was answering a very different question.
That's horrible. When you were down in the Amazon, were you around the pink dolphins?
So we were far out to sea.
Okay.
So what's really interesting about the Amazon River plume, so the Amazon River delivers more fresh water to the oceans than any other river. And I think all the other rivers combined, if I'm not mistaken.
Oh, wow.
And so that fresh water, because it's less dense than seawater, sits on the surface of the ocean once it gets out of the river mouth, and it goes thousands of miles offshore.
Really?
So you can be out, thousands of miles into the middle of the Atlantic Ocean, and it smells like an estuary.
Right.
And there's this layer which is like 10 meters deep, which is just fresh water.
Well, that's crazy.
All right, and full of all the freshwater microbes and things. So. And plays a huge role in like things like the iron cycle. So iron is really important for a lot of microbes. And so all of that iron comes from land essentially, and it gets basically put offshore in these big river plumes, and eventually it mixes in, but it is a huge influence on how the oceans work out there. So really fascinating stuff.
Yeah, that makes a lot of sense, because I think if I remember, I did an episode on the pink dolphins, and if I remember correctly, they think that because they're freshwater dolphins at this point, right? But they work their way in from the ocean, so probably just gradually adjusting to that, I guess, from saltwater to freshwater, because they were kind of in it anyways in that area. That's interesting. I didn't know that.
Yeah, I would guess there's a lot of, most of the material, like at least microbially, is kind of a one-way trip, if you will, from freshwater into marine. If you think of river flows, they just go down. But there are these mixing zones where, fresh and salt water mix, estuaries, and basically, sometimes offshore can act like an estuary because you've got this constant mixing of fresh and salt water when you've got a constant flow like that. So, but with animals, obviously they migrate, and they can go from the ocean inland. I mean, salmon, for example, So over time, animals will go into these environments that they find a particular advantage. They'll be successful there. They'll pass on their genes. They're fitter, if you will. And so then you get these establishment of populations like the pink dolphin, I would guess. I don't know a lot about pink dolphins, but that would be what I would assume has happened in the past.
I could tell you some cool stuff, but that'd have to be over a beer that I read about. But anyways, now the last thing is I want to go back to Palos Verdes because I'm sure when you were at school, the tide pools there were phenomenal. It was a kid's paradise, and I could walk from my house to where where Point Vicente was, right, and down in that area there, and there were so many. And as a kid, sea urchins to sea cucumbers to I would say starfish, but sea stars and all that. It was just this amazing aquarium that you could see so much in such a small spot. It was really, really special.
Oh, absolutely. Actually, true story. I lived about 3 blocks from Point Vicente when I was there. I lived in San Pedro at the end of Point Fermin.
Yeah.
And I used to go down there a fair bit at weekends just to go and check them out. And it wasn't really kind of my research focus, but it's always nice to see marine life in those tide pools. And bringing people in contact with them is totally critical. And I remember parking being an issue there. I remember parking lots being totally full and not being able to get down.
Yeah.
It was tremendous diversity of algae and invertebrates and some fish and things like that as well.
Oh, lots of fish too, because was Marineland there when you were there then? Probably.
I don't think so. I was there kind of in the early 2000s.
It was probably gone. So Marineland was there, but there are all these different places. We'd go down fishing. I could walk from my house, but away, not having to worry about the parking lot. And I could go in areas where not as many people were, but it was. I think about it now. It's that sense of wonderment. There was every time you went there, you saw something new. It was just so cool. It was really cool.
Yeah. Well, even if you don't see something new, if ever you're near a tide pool, with some care, okay, you can turn over rocks, and for sure you're going to see other invertebrates because many of them are just hiding. Like brittle stars, for example. Like I can tell you at a site we work at, the place looks like a whole bunch of barren rocks, maybe with some algae on them. But if you turn over those rocks, you'll find thousands and thousands of these tiny little brittle stars that move very fast to get away from you. They're used to their escaping predators, but rock pools are incredibly diverse. Now you have to be a little careful, particularly for listeners in Australia. I'm sure people are familiar. We have something called the blue-ringed octopus down there, which occurs in tide pools, and that's a really important one to avoid. It actually is very, very toxic. And if it bites you, it causes your respiratory failure, basically. But beyond that, just be careful with them. Don't handle things that look spiny, cone shelled. You want to avoid for obvious reasons. They're really toxic as well. But it's nice to look.
I want to tell you about something that's really cool. But first, let me set the stage for all you parents out there, new and old. Think about being so exhausted you can barely function. Now think about some of the chores you just want to do, especially when you're a new parent. Now how about this chore? Changing that stinky litter box. Yep, that one. Let's talk about Whisker's Litter Robot and how it eliminates that chore so you don't have to think about it. My daughter recently got her litter robot, and let me tell you, she's got a smile that's a mile wide. Life's busy, especially for new families. So, Whisker Litter Robot taking over the daily litter box duty? It's a breath of fresh air, literally. Maintain your cat's litter while focusing on your growing family and learning more about Whisker Litter Robot models and starter kits today to get set up before your new baby arrives. And take an additional $50 off bundles with code FORREAL when you shop whisker.com/forreal. Again, that's an additional $50 off bundles with code FORREAL at whisker.com/forreal.
Support for this podcast comes from Progressive, America's #1 motorcycle insurer. Did you know riders who switch and save with Progressive save nearly $200 per year? That's a whole new pair of riding gloves and more. Quote today, Progressive Casualty Insurance Company and Affiliates. National average 12-month savings of $197 by new customers surveyed who save with Progressive between October 2024 and September 2025. Potential savings will vary.
For real, 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 for real.
And we'll be right back.
As a pet lover, did you know your pet can benefit from the incredible healing power of stem cell therapy? Prep Therapy offers a groundbreaking solution taken right from your pet's own blood. Say goodbye to joint pain, arthritis, kidney and liver conditions, and more. They'll experience faster recovery, increased vitality, and extended quality of life without the need for surgery or harmful drugs. Put the wag back in their tail and discover PrEP therapy today at PrepVet.com. Save 10% with promo code FurRealPodcast, covered by most pet insurance.
So let's talk about your early days to moving forward with the sea stars. By the way, so help me on this. Sea star, starfish, they're the same thing, but it's the proper name is sea star. Is that what it is?
Well, yeah, no, I mean, the common names are either starfish or sea stars. We tend to avoid starfish just because they're not fish.
Right.
And I think there's some confusion about that. But, same can be said for sandfish, sea cucumbers. It's just starfish are kind of more common in our common language, if you will.
Right.
But, generally when we're going after common names, not the Latin names, we use sea star just to distinguish them a little bit better.
Right.
And even though they are actually they're more related to fish than other invertebrates as far as evolution goes, but they're still not fish. They're definitely invertebrates, although very weird invertebrates.
Okay, so let's go. So tell me how you got involved with it. And for anybody that's listening, if you didn't hear about the sea star, I'm going to say it right now because I don't want to be that guy, sea star wasting disease. But let's start from the beginning, how you got involved. And I kind of want to go year by year because I read a background on how what was originally thought and then your thought process changed. Let's take me down that journey. I'm really curious about it. And it's a fascinating story. It's a sad story at some level, but it's a fascinating story.
Yeah, no, it's definitely a story. In fact, 5 or 6 years ago during COVID, I almost made a podcast about it, just a limited series, because it kind of reads like a bit of a mystery novel, really.
Yeah.
So let's see, it was 2013. We had just actually published the first ever report of viruses in these sea urchins that we got from Hawaii that I mentioned earlier. And, I had a colleague at the time here at Cornell, Professor Drew Harvell, who's now retired and out of University of Washington. But I had been joking with her. I'm like, great, we got the tool. Wouldn't it be great if there's a mass mortality of echinoderms that happens tomorrow? We could apply these new tools to it. And be careful what you wish for, because literally within, I'd say about 2 weeks or 3 weeks after that, all of a sudden I get this email with 20 people attached to it saying, the sea stars are dying on the West Coast, and it's, there's photographs attached of all these gnarly looking sea stars and animals dying left, right, and center. And I was like, well, that's cool. We can, not cool for the animals, cool for us.
For us scientists, of course.
we can go and look at them and see, we can use this approach that we have to figure out if there's certain types of parasites associated with it. And initially I was excited. It wasn't something that we were funded to do, but and being a sort of early career faculty at Cornell, I was, keen to jump on anything which seemed like it might sort of be interesting or it might be high profile or whatever. And so I sort of stood back from the emails a little bit for a week or so, but it just seemed the volume of emails just coming in everywhere. All the public aquaria on the West Coast, like the Vancouver Aquarium, Seattle Aquarium, Monterey Bay Aquarium, Long Beach Aquarium, they were all reporting these, massive sea star deaths, not just in the wild, but also in the sea stars in their aquariums. And I was like, okay, well, let's, what do we need from this? What do we know about diseases of echinoderms, which at that time was nothing? What does a normal sea star look like what's its general biology? Because I didn't know that much about it. What are the sort of microbes that occur on a normal animal? And could we be able to distinguish what's disease compared to it? And of course, that wasn't known either because nobody had looked. But so we set about very quickly sending out instructions as to what tissues to take, what samples, how to prepare them, how to ship them back to me. A colleague, Drew Harvell, she actually put out a flyer which had the wrong address on it. It went to the building next door with all of them. So
Sounds like something I would do.
I was like, great, and also put my FedEx account number on there too, which is even better. So I spent a lot of money doing this. But basically, we had a lot of people out west, everyone from citizen scientists through veterinarians at the aquariums through to ecologists who are working at various institutions they sent us samples to Cornell in Ithaca, New York. We're about 7 hours away from the ocean. It's a great place to do this kind of work because there's no way anything that escapes from our labs can possibly go anywhere, right? Where, it's a great quarantine facility, which feel in many different ways, let's put it that way. But anyway, so we got these packages of, frozen, semi-frozen, sometimes not even frozen, dead and dying sea stars. And, we asked for healthy sea stars too, to basically compare them to, because we didn't have any frame of reference.
Sure.
As it turns out, we got like 90% dead or dying sea stars and about 10% healthy ones, because I think, everyone is concerned when you see a big outbreak like this. They don't want to get rid of or destroy the healthy ones, right? They're the survivors, and they might go on to spawn a new population. But without those comparative samples, it's really, really hard. So we had a few comparative samples to do. And, going into it, we took a sort of slightly, I'm going to admit, it was slightly biased. I was working with viruses, and I was interested to see if it was a virus. And so we found, guess what, a virus through a comparative approach.
Right.
And, when the initial data came in, it was a little bit of squinting going on, looking at the data, saying, boy, these things don't look that different between the diseased and the healthy ones. We looked at bacteria as well. We worked with veterinarians who were sort of looking for other bigger things, like big parasites, multicellular parasites. They didn't see anything either. And but really, the only thing that kind of sort of made sort of some sort of difference between healthy and disease was this thing called a densovirus. And it happens to have been the same virus or same family of viruses that we had recovered from these urchins in Hawaii, which was, again, a novel kind of observation. These types of viruses were only previously seen in things like cockroaches. And, there's one that occurs in humans. It's a parvovirus that causes slap cheek disease in infants. And so it was kind of like, wow, okay, so maybe we'll look at this. And so it gave us sort of a candidate. It gave us an idea of what might be going on through this analysis. And so the next step for it is, obviously when you see a microbe associated with a disease, like only the diseased ones or mostly the diseased ones, it doesn't necessarily mean that it's actually causing the disease. In fact, when you damage or you get sick or whatever, you get a wound, you're always going to get a ton of microbes in there.
Sure.
That are just opportunistically, eating the materials which are being released from the wounds or from the sick animals, okay? It's expected. These things are called saprobes if they're eating dead tissue, or opportunists if they're eating, be able to get into the tissues and cause, and basically consume the materials within the tissue. So it wasn't just enough to go in and see this virus. We needed sort of experimental evidence to see that it was actually causing the disease. And so we fortunately were able to get some sea stars into the lab in Ithaca. These were tiny little sunflower stars that we got from an aquarium. I think it was the Seattle Aquarium at the time. We were able to get also we had the diseased tissue that people had sent us. And so what we did is we actually ground up that diseased tissue. We filtered out all of the stuff that was like, bigger than a virus, right? It also included all the stuff that was smaller than a virus too, but we sort of ignored that. We did a control where we took those viruses, we boiled them to kill them, because boiling does a good job at destroying viruses. And we injected them into the sea stars, and nothing happened, okay, for that first experiment. And so it's kind of like a big bummer when that happens, right? It's like, boy, we get this evidence.
No, we don't.
Right, exactly. It's like, but it isn't doing anything. Wait a minute. So what is it we're looking at? And so we then, took a break from it. We tried again, another experiment, and it didn't work again. We were sort of questions swirling as to maybe it's not a virus, maybe it's some other microbe, but then there really wasn't anything that we could identify by comparing, healthy and diseased animals. And so we started to then, fortunately, Drew Harvell and a couple of scientists from USGS out in Port Townsend in Washington State, they had a great system, an aquarium system for maintaining larger animals, these larger sunflower stars.
For everybody who's listening that don't know, and the only reason I know it because I was doing homework for it, tell everybody what a sunflower star is and how big they're big. So tell them a little bit about that.
They are huge. So these are sea stars that don't look like your classic like 5-arm sea stars. These are giant. They can grow up to like 1 meter across. and they have 24 arms or more.
That.
They are, most of the animal is actually just the center part of it, what we call the central disc. And then you get these little arms that come out the sides. They are incredibly voracious predators. They eat everything out there in the ocean, like, everything from their natural food, which is sea urchins and little mollusks and that kind of thing that they pry off the seafloor. But they'll also eat other things. Like, for example, there's a squid fishery near Santa Cruz in California that discards all the squid beaks after, so it's the part you can't eat. They would find the Pycnopodia basically eating those.
There's a catfish or pig of the sea.
Oh, yeah. They're kind of gross. I mean, everybody thinks sea stars are beautiful, iconic animals of the ocean. These things are nasty. They're just like brutal killers of everything out there. But anyway, and the sunflower star is kind of interesting because it is gelatinous, right? It actually doesn't have a lot of hard bits in it. Okay.
Right. Oh, that's interesting.
Pretty soft animal. And there were thousands. I want to say thousands. There were billions of them up in the Salish Sea. So Puget Sound down to Seattle, and they became such a problem in the decade prior to that with fishers, so people that put down crab pots and that, they would clog up their crab pots by getting in there and stealing their bait, and people would basically chop them in half and send them, back in the ocean, not knowing, of course, that becomes 2 starfish when you do that. But basically, these things they're huge. They're really important for the ecosystem because they do keep in check things like urchins, and urchins graze out kelp.
Right. And
when you don't have the sea stars, the urchins go crazy, and you don't have kelp. Right. So it's called a keystone species in that regard. But they are important. Don't get me wrong. But they are little animals. They're really nasty little animals.
Brutal.
Not little. They're pretty big.
Yeah.
But so they, and this is one of the species that was the most heavily affected by this condition, sea star wasting disease, if you want to call it a disease.
Are they endangered now?
So their populations were hit very, very hard in 2013 and beyond. They are on the IUCN Red List now. They got it listed there. I don't know that there's a specific definition for endangered for marine invertebrates because, we don't have ways of surveying where they are, right?
Right, yeah.
So, or putting some sort of bounds or quantitative variables on them. But what I can say is that for a period of several years back after this disease, they were basically functionally extirpated or extinct in the whole of the Salish Sea. The divers weren't seeing many of them at all. And basically it went all the way down to really, really deep waters that they were just gone. I mean, these things go from the intertidal all the way down to, several hundred meters deep, and they just couldn't find them. So, I would say, if you want to use the word endangered, they were heavily reduced to the point where there was concerns about them being able to repopulate.
Wow.
Put it that way.
Okay.
Yeah. But they've been doing some efforts since the disease up in Friday Harbor Labs to try and raise them in aquariums and put sort of brood stock or juveniles out in the intertidal. There's a lot of efforts at restocking which are underway. And I don't know the success of those as yet, particularly in the wild. I know they've been successful in raising them in the lab, but generally what happens when you release, farm-raised, aquarium-raised animals is they get picked off by predators as soon as they get out in the ocean because
they're not used to hiding from them or doing whatever.
Exactly, or the microbes that are out there are different from the microbes in your aquarium.
Right.
So, but it's good that they're trying.
So you get these sunflowers, and you get them. So what happens then?
So once we get the sunflower stars, in terms of like what we did with them experimentally. So this is up in USGS, and this is work done by Drew Harvell and others. This wasn't my work at all, although it was part of the publication we put out in 2014. So basically, they ground up an arm of a diseased sea star, filtered out everything, similar to what we did in Ithaca, injected it in there, and then looked for the appearance of like this disease. In this case, it was looking at these sort of lesions, these white spots in their body wall falling apart, right? And they did that for the first time, didn't really work. Second time didn't really work either. Third time, it totally worked. Third time, they had animals that were curling their limbs. You had these lesions appearing on their surface, and it was totally related statistically to the injection of this virus-sized material into the animals. And so that was satisfying what we call Koch's postulates, which is the set of criteria that Robert Koch came up with in the late 1800s that basically proves beyond reasonable doubt that a microbe is associated with the disease. And so we were very excited by those results. We presented a paper. We published it in a pretty high-profile journal, the Proceedings of the National Academy of Sciences, in 2014. Lots and lots of news coverage. I can't remember how many interviews I did for the media, CNN, Fox, you name it, everyone. It was translated into, I think we said 28 languages or something as well. It was pretty cool.
That's cool.
And so, it was a great story. The only problem is, and this is kind of the funny part of the whole thing, we then, asked National Science Foundation for further funding to understand more about, how the microbes, how the virus actually generates the disease process, something called pathogenesis, and how it relates to environmental variation. That we secured that about 6 months later after that paper came out. And then we could not, for the life of us, make that disease happen again. Over and over and over. We tried 6, 8 times, and it was oh my goodness, what's going on here? And we sort of revisited different hypotheses, maybe it wasn't this virus, maybe it was some sort of environmental variation that was induced in the aquarium system that we weren't too sure what it was, but we started to look at long-term data monitoring sets from oceanographic buoys and things, and looking at temperature swings that occurred weirdly just before this disease started, but we don't really have a firm time when it actually started in 2013. We started to question what sea star wasting disease really was. Was it a disease, period? And it was mainly because, it was presented, or when we first went into it, we thought, oh, this is a brand new thing that we've never seen before. All the sea stars are dying en masse, right? And we looked through the literature, and we couldn't really find any good descriptions of it at all. It was, maybe here and there was a starfish died and their guts were hanging out, but there really wasn't sort of a mass mortality setting. But then we started to become alert to other literature in conference proceedings and historical literature where they use different words to describe sea stars and wasting, oldie language, right? And it turns out that this condition had been around since the late 1800s.
Really?
And it was okay, so this is not a novel thing that started. And so maybe something's changed in the environment that's made this thing pop out, and maybe it's causing that. And so we tried everything. We tried everything experimentally. We tried temperature. True story. You put aquarium heaters into an aquarium thinking that it'll kill everything, whatever. You come back the next day, the starfish will guarantee be trying to eat those heaters, regardless of how hot they are.
Yeah, that's funny.
They behave or air stones. That's the other thing. They love to eat air stones, which is really hard. Makes it hard to work with.
Yeah.
But we couldn't find any answer at all to it. And, started to revisit the viruses that we were looking at. And, what we realized pretty quickly was that there's a whole suite of different viruses which were actually being lumped in together into one virus that we had identified, the sea star-associated densovirus. And when we started to dissect those different viruses, we found there was absolutely no relationship at all. Okay, there was just really flat line between control and healthy. It's just because we had lumped them in, it made it look as though they were more abundant. And in fact, the virus was, when we already knew this, was in a lot of the healthy animals, totally healthy animals. So it probably wasn't the cause of that. So we early on came to the conclusion we were wrong.
Which I think it's awesome that you admit it, because that would be really hard to do. You're a scientist guy, we got it, and all of a sudden, sorry, we're wrong.
Oh no, totally. Every scientist will try and defend their research, right? And I certainly did that. But after a while, after hammering at something for a long time, if it's just not working, if it sort of falls apart from a data analysis perspective, you just, in this case, we were totally wrong. And we have two routes for basically going forward from that. You either retract the paper that you originally published. So that means formally withdrawing it from the journal. As it turns out, the data on which was reported in that, our interpretation was actually correct based upon that data. So we didn't end up doing that. But then I published, I don't know how many, 5 papers or something like that after saying, after a while, just saying that was not correct. It was not correct. I had to change the Wikipedia page because it still had it listed as the cause of it, And so, but of course, the big question after that is always, what is causing it, right? People love answers.
Right.
We love answers. The scientists, we want some explanation for it. And so we tried a variety of different experiments, like I said, with temperature. We tried adding different types of bacteria, including Vibrio, one that we isolated in the lab. We tried making them upset with harsh language, that kind of thing. And nothing we did could basically make these sea stars die. And so we had a final ditch effort in 2018. We went out to, it was Long Marine Lab at the UC Santa Cruz, and we were going to try this really complicated experiment where basically we were going to make them really hot, then really cold, then really hot again, because that seemed to correspond with the data that we were seeing from oceanographic buoys. Very, very difficult to do, by the way. It's not like turning on a tap and doing that. You have to have these big, expensive chillers and heat transducers and things like that. But we were able to do it. And so we went out, we collected, I think it was 25 sea stars from near Natural Arches in Santa Cruz, put them into a sea table to let them sit and acclimate to being in the lab before we started the experiment. We went back, I came back to Ithaca, my team and I, and about a week later, all of the starfish started to have sea star wasting disease. And it was like, wait a minute, we haven't seen this in a while. We're not even trying to cause this disease. What's going on? And so we had some collaborators at UC Santa Cruz. Lauren Schiebelhet's one of the big scientists who works on this out there. They harvested all the tissues. We asked if there was anything weird going on at the time. Nope, there was really not much change going on in the sea tables. It wasn't exceptionally hot or cold out there. And so it was like, okay, well, that kind of blows our experiment. But now we know that the disease is around. We're going to try and hit it with other types of things. So we went out, we tested everything from adding, again, the tissue homogenates. We tried turning down the aquarium flow rates to the point where, they weren't getting much replenishment in the water compared to really, really high ones. We tried rubbing the sea stars against each other in case there was some physical damage that was causing it.
Yeah.
I can't remember what the fourth one is that we did, but basically everything we did caused sea star wasting. So we went from the situation where nothing caused sea star wasting to everything caused.
Everything does.
So classic marine ecology, disease situation, right? It doesn't inform us at all.
Right.
And so it was kind of back to the drawing board. And one of the things we did during that was to take samples looking at the microbes which live on their surfaces as they went from completely healthy to completely diseased. And we got the data back. It takes a little while to do this work. It's sequencing, DNA sequencing is fast, but it's not that fast. We started to look at the data, and we realized that, it's pretty boring. There's a ton of different types of bacteria that you get on wounds normally, and things like Vibrio, which is a totally fast-growing bacteria that grows on any and all carbon in the ocean, pretty much. But then we noticed that there was these anaerobic bacteria that started to appear as the animals started to get sick. And we were thinking, wait a minute, why are there anaerobic bacteria? These animals are living in the intertidal zone. They're being bubbled. There's no oxygen. There's plenty of oxygen. And these are bacteria that cannot live in oxygen, but they were there.
Right. And
so it kind of spurred this idea, well, wait a minute, is this some kind of hypoxia event that we're seeing? There's no hypoxia in the water column, right?
Yeah.
There's tons of air out there. So then we started to look at different patterns of where this thing was occurring in the ocean. So basically, some places when it was really hot, they were dying. There was a distinct correlation there. Some places where it was really cold, particularly along the coast of Oregon, it negatively correlated with temperature. But it sort of got us thinking, what possible linkages? This is this. I remember sitting on a 29-hour flight from Australia back to the U.S. and virtually the whole flight thinking about what could this possibly be. It was a real brain scratcher, this one. But then I was thinking, could it be that you have more like algae in the water, and that causes, it produces a lot of carbon, so it produces a lot of sugars and proteins and amino acids and all the stuff that bacteria love to consume. They, in turn, on the surfaces of the animals are going nuts, basically. And when they do that, they use up all the oxygen, this very, very fine layer on their surfaces. And then that, in turn, might starve the starfish of oxygen. They're suffocating. And so that would fit with the higher temperatures because obviously warmer water has less oxygen in it. So you boil a kettle and all the oxygen disappears, right? You could also fit with the cold water as well because that's where you have upwelling, the movement of deep water onto the shore. That in turn brings all these nutrients with it, stimulates algae, and that in turn stimulates bacteria on the surfaces of animals. And so we then went to Bodega Bay the year after that and basically did experiments with organic material, algal exudates, and whatever, and it actually worked. We could induce this condition with algae, with basically carbon. And so we put out this paper shortly after the pandemic, or during the pandemic, I might say. It was actually January the 6th, 2021, that basically talked about this phenomena of boundary layer microbes which are driving this disease. Wasn't met with a lot of popularity because it basically said it wasn't a disease that was transmissible. It wasn't a disease that could move from animal to animal. It was driven by this sort of process on their surface.
Environment.
And because there was a lot of investment in disease ecology research, and particularly in identifying the cause, it was kind of unpalatable, right? And we looked at things like the rugosity of the starfish. So when you get more sort of corrugated surfaces, there's bigger boundary layers, and those were the starfish that died. And looked at something called stable isotopes as well, which gives you a good indication of the oxygen conditions on their surfaces. And that gave us some evidence that there was something hypoxic happening there as well. But it really didn't get a lot of attention. But that was kind of the final say in the whole sea star wasting investigation for me, because literally for 8 years, I had been scratching my head trying to figure it out and tested anything and everything, trying to link together very, very different environments, different species, 20-something species affected, into sort of a solitary phenomenon that was causing all of it. And so that was the end of it for me.
I've got several questions for you. So here's the thought I had when we were talking about this. I'm sure you had that thought. If I'm a believer in climate change and all that kind of stuff, so even if your water is colder up in Oregon versus Santa Cruz, even Santa Cruz, the water's cold, but you go farther south, because it went all the way down to Baja, right? All the way, right?
Yep, and up to Alaska.
But and again, I'm following you with the algae in that too. So if the temperature is warmed up just a little bit on average over the year, even if you're used to cold water, if it's a little bit warmer, is that playing a part of it, or is it possibly, I guess, at some level? But I'm assuming that has to do with algae growth and all that, the warmer it is and all that. Is that kind of what's going on? I mean, based on your hypotheses?
I mean, if you want to talk about averages over decades, then for sure, the oceans are definitely warming up. There's no question about it. Like we see range shifts in tropical organisms into temperate waters all the time.
Sure.
And, you go to Southern California and they get some tropical cyanobacteria there that wasn't there when I was there 20-something years ago.
Right.
And so it's really evident that the oceans are changing. But in this situation, what's really interesting is that the disease took off during a cold period. And basically, it seemed to prefer colder. So you might have heard of like the blob that happened in the Pacific Ocean
I remember that.
2010. So that was the year after this happened. So this was well before that. So while we can't negate the fact that animals are probably stressed due to increasing temperatures in general on average,
right?
This thing didn't correspond with it at all, and it seemed to it was definitely it started in cold conditions. And so, animal stress, the microbe which is infecting them, you put the two together and there's a sort of magic combination that results in disease. And so anything that stresses the animal obviously is going to increase its susceptibility to the pathogen.
Sure.
And that might have been a play over sort of decadal scale. So this thing might have come in, whatever caused it, a while ago, and it's just because of this increased temperature, on average, that it's become more abundant in the ocean, and the animals becoming more susceptible. But in the 2013 event, we just don't know. I would argue against it, but yeah.
So now, though, because it's kind of not, it's still around, but is it still around or is it gone now? Because I know it was a big deal and now it's gotten better somehow. So walk me through that. I don't understand it.
I would say, 2013, 2014 was a really bad year. 2015 was also, it was noticeable. It went away, and it sort of became very low level. And then beginning in like 2018, 2019, there were a couple little outbreaks here and there. Again, very low level. And till this day, it's still kind of, when it occurs in a relatively low amount. Citizen community scientists out west, they'll report that there's sort of a few Pycnopodia or other species that are infected or affected, but there's not like the massive outbreak that happened in 2013 that seemed to affect like billions of them,
Right.
And so, things have been quiet for a few years on the sea star front, at least on the research front, until August of last year, when another group of investigators, including Professor Harvell, who's retired from Cornell, they reported that there was another type of bacterium which was responsible for it, and this was based on looking at their blood, their coelomic fluid, this water that circulates around their bodies. And to date, that's the most convincing kind of case for a cause of it, I would argue. There are some caveats with it, though. We don't find that bacterium back in 2013, so I still got a freezer full of dead starfish, and we surveyed them, and we didn't see it there. It's also a little unclear whether the sort of, if you want to put disease symptoms, symptoms are self-reported, so starfish can't self-report, but we call them disease signs. But for ease, I'll just say symptoms. So the symptoms that they're reporting with the current experiments that they're doing are basically their arms are twisting, which starfish do when they're grumpy. And they also don't have veterinary tissue level sort of analysis, right. Histopathology, we call it. And it's going to be really interesting in the next, period to see whether that actually comes out to support it or not. But
so it's still kind of a mystery.
It is. I think that there's always room for doubt in science. And I will say, the authors on that paper, Alyssa Gaiman and Melanie Prentice and others who are up in Canada, did a really amazing job with what they did. I really respect the work that they do. But these are complicated systems, right? Complicated animal. And I really, really hope that they're able to pursue further investigation and find if this is actually it, or if it's not it, then what else? Caught in that loop of wanting to know the answer, but everything coming up, as a roadblock. So. For real, for real, for real, for real,
for real, for real, for real, for real. And we'll be right back.
As a pet lover, did you know your pet can benefit from the incredible healing power of stem cell therapy? Prep Therapy offers a groundbreaking solution taken right from your pet's own blood. Say goodbye to joint pain, arthritis, kidney and liver conditions, and more. They'll experience faster recovery, increased vitality, and extended quality of life without the need for surgery or harmful drugs. Put the wag back in their tail and discover Prep Therapy today at PrepVet.com. Save 10% with promo code FurRealPodcast, covered by most pet insurance.
I want to get into some of the stuff you're doing now, but I got a couple other questions I thought about when I was reading that I don't really understand. First off, this is a little bit different, and then I'll get to a couple of things I'm curious about. Well, I'm curious about it all, but one of the names that you originally were going to call was asteroid idiopathic, something or another. Mm-hmm. So having, as you know, I work in the medical world, so I know what idiopathic is. And for listeners, you can kind of give them the definition. I'm curious where that name came for you when you were doing that. Were you thinking the system was working against itself or something? I'm an idiot anyway, so we're good with that one. That's why I know the name, because it feels my personality.
Yeah, I do not claim to be a good namer of conditions.
I love the name personally because I'm an idiopathic. Well, that's kind of curious. Is a starfish working against itself?
So, I mean, like if I said to you a disease, like there's a disease in an animal, what do you think of that cause of that disease is?
I'm just germs.
Germs, right? A microbe, an infectious microbe. Right?
Yeah.
So, but diseases are actually a lot wider than that. I mean, alcoholism's a disease. Arthritis is a disease, and those are not microbial diseases at all. And so, I went down the rabbit hole pretty heavily of like trying to distinguish between a disease, a condition, a symptom, a syndrome, you name it, right? A disorder. The fact that we couldn't find a microbe which was associated with this distinctly at the time sort of led me to rename it from sea star wasting disease, because that immediately in many people's minds implied that it was an infectious disease when we had zero evidence at the time we named it of that.
Right.
To asteroid idiopathic, which basically means no distinct cause, right? Probably a more technical term for it, but wasting syndrome, right? And the wasting part just means that the animals are falling apart, which you have deer wasting disease and other things. It's just kind of a catch-all.
Sure.
So we renamed it in the literature that it didn't catch because everybody still called it sea star wasting disease. I actually trimmed off the disease part. I call it sea star wasting, mainly because I think that
disease is not a good definition. I get it totally.
Disease it's amazing how it inspires this idea that it is something caused by a microbe,
right?
Subconsciously. And I'm still not 100% convinced, although, I think that what the recent work on Vibrio pectinicida points to it, right? But I'm still not totally convinced that it's a microbial disease, or at least not an infectious disease caused by microbes.
So the other two things that I came across when I was reading, and I don't know why I'm not familiar with it, is RNA viruses and DNA viruses. I don't know what they are. I know what RNA is. I know what DNA is, but I don't associate virus with that. So can you just give me a little bit of thumbnail sketch of what that is?
Oh yeah, for sure. So, all cellular life on the planet has a single ancestor, right?
Right. Sure.
Long time ago, probably a single-celled organism bifurcated into two, and became all the life that we know, cellular life on the planet. So viruses, on the other hand, probably came from multiple different origins, right? And some of those viruses kept DNA as their core information storage, right? So things like herpes viruses, they have double-stranded DNA in their genome. That's a common one. For the densovirus, that was a single-stranded DNA virus. And so those DNA, their genes essentially are in DNA. But then there's this whole other group of viruses, which is part of this realm called riboviria, which basically only has RNA in their viruses, in their virions. And what's really interesting about those is that, so normally when any animal or any cell starts to create like genes or transcribe genes, it goes from DNA into RNA and then it goes into protein. And that's called the central dogma. With these RNA viruses, they short circuit it. So they basically just go from an RNA strand straight into a protein, and they start to create new copies of themselves, cause pathology, et cetera. A good example of an RNA virus is a coronavirus. Unfortunately, COVID is the coronavirus that caused SARS-CoV-2 RNA virus.
Right.
So, but there's also some weird viruses. Like you get these giant viruses that look nothing like we've ever seen before. They're the size of small, like protists, things like fungi, right? Tiny little DNA strand in them, but the huge capsid, the protein part on the outside, they probably came about from some other origin altogether. We don't really know. So, but yeah, DNA viruses and RNA viruses just refers to the type of either DNA or RNA, which is in their genome. And it's just two broadly different types of viruses. You get two ones that have both, which is really interesting.
Yeah.
Fascinating.
So I'm going to go back to what you've been doing, because you've moved on from the starfish and you're doing that. Let's talk about that and what you're doing. Because one of the things I read, and I don't know if it was from our conversation that you and I had beforehand, We almost have environmental diseases now that are caused by either the climate change and all this other stuff that's going on. And I'm assuming that that somehow relates to what you're doing now. Am I right on that?
Well, we think so.
Okay.
That's a hypothesis that we're testing at the moment.
Okay.
So basically so cast your mind back to the early 1980s or look up a history book if you're younger than whatever that is, 30 years old. So basically in the Caribbean back in the 1980s, all of this one type of sea urchin called Diadema antillarum died off spectacularly. All right. So prior to that, the reefs were in, not so great shape, but at least they had healthy corals and they had a sort of small amount of algae out there. And then in about 1982, 1983, 1984, all the urchins got wiped out. Algae went crazy, smothered all the corals, and you can see the result of it today if you go down there. There's very poor coral reefs. So we never worked out the cause of that at all. And the urchins basically didn't rebound for, the last 30 years. And so there were about 5% of them were still there in about 2021 compared to what there were before 1983. And so in 2022, all of a sudden, those remaining urchins, boom, they started to die off again. And we went down there very quickly, recognizing that this is, another important thing. Why is it happening? This is sort of at the tail end of COVID lockdowns, and cruise ship traffic had started again for the first time in a few years. And not that they're the cause of it. I don't want to infer that, but just to point out that tourism was happening again. And so we very quickly worked with our collaborators across the Caribbean, 13 different countries down there. We worked to get specimens from the Saba, which is in the Kingdom of Netherlands. It's a tiny little island south of St. Martin, St. Thomas, U.S. Virgin Islands. And we looked, and we were faced with kind of a similar situation as sea stars. It was like, wow, nothing really looks that different again. And I'm like, oh no, not again, right? But then I looked at the gene transcription of the animals, the healthy and diseased animals, and picked out a ciliate, which is a type of protist. It's like a type of fungi, eukaryotic organism, that shouldn't have been there. And I was like, well, that's weird. And so I remember seeing the data on my computer, DNA data, and running down to the lab to look at some specimens under the microscope. And I was like, I wonder if there's like ciliates. And sort of moving towards the microscope with my eyes, I was like, oh yeah, that's it, right? Down the microscope is absolutely loaded with these ciliates. But again, it was like, does this cause disease? Is it just something which is eating decaying tissue? So we're in the really good fortune of having collaborators in Florida who were able to culture this microbe immediately as soon as it appeared there. Bill Sharp down at U.S. Fish and Wildlife, or Florida State Fish and Wildlife, sorry. And then, so we got a culture of it. We then had really good luck that the Florida Aquarium was working with somebody in Tampa Bay, basically Apollo Beach, to get like naive urchins that were raised in an aquarium. We knew that they were totally clean of any potential pathogens. And then we also had USF, University of South Florida, right there, and worked with some collaborators down there. Maya Breitbart, in particular, is my longtime collaborator. And we did this experiment, and it worked straight off the bat. It was like, wow, 60% of the animals died when you add the ciliate to it, and compared to controls. And so that was fast. It was like 6 weeks, wow. Literally 6 weeks from the time we first were alerted to it to the time that we had proven or demonstrated Koch's postulates. And this ciliate, we then went out into the field and we looked at different samples that we collected around the Caribbean from coral surfaces, because by that stage the urchins were all gone, they'd all died. It was really dramatic when it happened. There were no urchins for about a year or two after that. And to our surprise, we actually found the same ciliate on the surfaces of some corals there. We also found it in sponges a little bit, and also on the surfaces of a boat hull. And so we sort of have started this project to look at, okay, it's there. What are the environmental drivers for it to emerge into urchins? All right. And so we have funding from the National Science Foundation. Thank you. And basically, we started a time series in September of last year to look at all sorts of environmental variables in concert with where the ciliate is in St. Thomas. to figure out, does anything correspond with it? Are there any obvious sort of blips or anything which might explain why this thing might emerge? And, the urchins have rebounded a little bit. They're there, they're big. And we were sort of thinking, well, maybe this won't happen again, right? So January rolls around this year, and of course it happens again. And so we had lots of data leading up to it and lots of data after that we're currently analyzing. So it's a super exciting field, and it's we have hypotheses that we're addressing right now through that I can't say definitively what the cause of that emergence is, but I will say, just like sea star wasting disease, it does seem to be related to algae to a certain extent.
And that's what a question I have is that, and again, we may have talked a little bit about this. It seems to me as water is getting warmer, there's probably more algae out. You got algae bloom and all this other stuff. So it's got to be affecting the oxygen in the water at some level. So is the oxygen level going down? Is it measurably going down? And how is that affecting marine life across the board? Is it really? Are you guys starting to see a bunch of changes that way?
Oh, for sure. Yeah, no, definitely. Deoxygenation of the ocean is something which has been documented for the last several decades, and it's only recently become really prominent in our literature that we read. There's Curtis Deutsch, who's at Princeton, does a lot of work to look at its impacts upon like zooplankton in the ocean, for example. And it turns out that, as the oceans are warming, it's losing, and I can't give you a percentage off the top of my head, but it is losing enough oxygen that's becoming harder and harder for animals to breathe, and pushing to the limits the physiological capabilities of some types of animals and zooplankton, tiny little animals, to actually survive. In context of the urchins in the Caribbean, once again, we're faced with that situation where, we don't see any distinct, big temperature pulses, which corresponded with it at the time. But we can say that, things like frequent storm events, we know that storms are becoming more frequent down there.
Right.
Rainfall is more in places that it shouldn't be rainy, and so runoff is more. That might be related to this, but we don't know. What's really interesting, though, and this is something we still are trying to get our handle on, this microbe that we know causes disease in urchins suddenly appeared in the Mediterranean about, it was 3-4 months after it was in the Caribbean. And then about a month later, it moved through there into the Red Sea, through the Suez Canal into the Red Sea, around the Arabian Peninsula, and then down into West Africa. And it looks as though it is it's moving against current systems. We don't know, the obvious answer would be boat traffic, right? But we have yet to address that hypothesis fully. We don't have any data to support that. But, when you see that kind of change across the board out there, it certainly suggests that there's probably a combination of new introduced things and then bizarre weather phenomenon or increased temperatures that are happening around the ocean that are influencing it. So it's something that we hope to have answers for in the near future through our project.
You mentioned something else, and we'll kind of finish this up because I know you're busy, but you had mentioned something earlier, and I wanted to ask you about this. It's about citizen scientists. Okay, so I know you mentioned it. I find recently I was talking to somebody when we recorded, and it's a guy that's an expert on praying mantis, and was really, those things are fascinating. But anyways, he discovered a new one, but he found one but wasn't sure, and a citizen scientist found that one and sent it to him, and that's how he found it. Can you kind of get into the importance of kids, everybody that really, if they're out there being, the doctors, Doctor Who or whatever they want to be, how important that is, and to you and to the rest of the world?
Absolutely vital. Scientists, people that are basically going out into the field to monitor locations, we can only have a set of eyes on it for a short amount of time. And, generally speaking, we don't have the geographic coverage that, is possible to look at these big disease events or, notice when animals are dying or what species are there, as you just gave the example. And so citizen or community scientists who are making these observations, who report their findings on things like iNaturalist and other platforms, or through something called AGRA, AGGRA, Atlantic and Gulf Rapid Reef Assessment, I think it's called. incredibly vital. We wouldn't have any clue as to how extensive this stuff is or timing of it as well if it weren't for community scientists making those observations. So, I'll give you an example of the importance of community scientists. Jan Hewson is a diver. He's a community scientist who lives up in Whidbey Island in Washington State. Every single week, he was diving at the same location, which happened to be where there was a lot of sea stars, and also where there was a data buoy nearby with chlorophyll and temperature and salinity and all that. So he reported his observations every day by email, or every time he went diving by email, about the occurrence of sea star wasting. And we could sort of estimate, was it intense? Was it just a few at the time? And by doing that, we were able to line up exactly the pattern over the year when this occurred and relate it to chlorophyll and relate it to temperature. And we wouldn't have a clue about that. Right. We can't be out there all the time doing it. And so Jan is like a real superstar for doing that. And of course he was doing it recreationally. He was just doing it for fun. But he's actually co-author on one of our publications that came out, because it's like, wow, your data is super valuable. But, for all the citizen or community scientists out there, know that your efforts are totally valuable. Absolutely. Record it. If you don't record it, it didn't happen, right? It's kind of like in science. If we don't publish it, it didn't happen. So, if you're able to see something, if you see something weird or unusual, find the platform and basically report that on its occurrence. And we thank you very, very much for doing that.
So if people are in whatever part of the world where the urchin thing's going on or the sea stars, is there a place that you would recommend that they send that information to? I'm thinking about like the junior Sherlock Holmes of the scientific world, right? They said, look, this is cool. And do you know what I love about this? Is what a great way to get kids involved that are saying, let's go out and check out whatever. And they're involved, and that's a really fun thing to do. It's exciting. It's an adventure.
Oh yeah, for sure. I mean, you can be part of a wider investigation and be part of the solution, right?
Yeah.
No, I'd say I know iNaturalist is got good coverage around the world, but there's also some regional sites that you can find, particularly in the Caribbean. This Atlantic and Gulf Rapid Reef Assessment (AGRA) runs a really good sort of community science reporting portal, particularly for divers and things, which is very helpful. You can, for listeners who are at other countries around the world, there might be national reporting databases or something, community science databases. So I'm not familiar with those, but iNaturalist I know is available in many parts of the world for doing that. But yeah, no, definitely become involved. Be a disease detective.
Yeah, I think it'd be cool.
Be a part of it, because and your observations are incredibly valuable. Just know that.
I wonder if IUCN gets people sending stuff into them. I'm curious about that. I bet they do, probably at some level.
IUCN?
Yeah.
I actually don't know the organization that well, but I know that they are fairly active as far as activism about endangered species and diseases. So, and you know what? If you do see something and there's no IUCN or iNaturalist or whatever, find your local university,
right,
your local college, and let people know.
Cool.
Just send a blank email to them, because they should be able to direct you in the right direction there.
I don't know this. I probably should have known. Do you do any social media at all where people can find some of the stuff you're doing? I'm guessing you don't. For some reason.
Well, I have my personal social media. No, I'm on Blue Sky. You can find me there, which I keep for mostly my research stuff, and then through Cornell CAL's College of Agriculture and Life Sciences, we have things that we're promoting, as well as this thing called Cornell Oceans, which is this new initiative at Cornell that we're trying to build marine science on campus. We have a social media presence as well. I'll see if I can find the links for you.
Well, what I'll do for you guys that are listening, if you're hearing this now, it'll be in the description. I'll get it from you, and then we'll put it in the description, and then we'll put it there. So we've come to that part of time which I know you were desperately waiting to hear this really funny joke I'm going to say. I know you really will. I know you wanted me to do it earlier, but I've saved it at the end, so it's kind of like dessert at the end of the meal. So, but for everybody else who's out listening, if you've listened to my episodes before, I always have a silly animal joke. But as I always say, if you want to be on the show if you've got grandkids and you want them on, nieces, nephews, or your kids, if you email me at mkyleforrealpodcast.com, gladly, I will tell you the guests would be thrilled not to have to tell one or hear mine. And I would gladly have you on. And if you've listened a lot, you know that I've had different guests on and do it. So again, it's mkyleforrealpodcast.com. So here's my joke for the day. Let's see if I can remember it. So why, you're going to die. Why did the starfish blush?
I don't know.
Because he saw the ocean's bottom. I knew he was going to love this one.
That's a beautiful one. I'll have to borrow that one.
Oh, yeah. I'll bet you'll use that all the time when you're out at the bar. Let's break it, guys. Anyways, first off, thanks so much for being on the show. I really appreciate it. I love this stuff. So this is great, and you're doing great stuff, so I really appreciate it.
Thank you for having me. It's been great. Appreciate it. And
now you have a great joke, too. Anyways.
Yeah, and thanks for the listeners for tuning in and sticking with it, too.
Yeah, well, I know they've already hung up at this point. But anyways, but for you guys that are listening, for me, please keep following, listening, pass the word out. Tell people about this episode. You know, without you guys, people don't find about it. But and the other thing for me personally is, it's for me, if the more I grow, the more I get opportunities to have great guests like Ian. So please keep following. But the other thing is always be kind to animals. And seriously, if you're an animal lover, you're a treat dealer. So, but really, lastly, from the bottom of my heart, thank you so much for listening. For real.
That concludes this episode of For Real with your host, Mark Kyle. For previous and future episodes of For Real, listen via all major podcast streaming sites.